Welcome back to the deep dive. Today we're starting with, well, a huge ecological issue, really, but it's driven by microscopic life.
That's right. Think about cows, sheep, goats, all those ruminants.
Billions of them. And inside their digestive systems, something remarkable is happening. They're producing about, 200 million metric tons of methane gas every year?
Yeah, 200 million metric tons. And you have to remember, methane is potent stuff, much more so than CO2, maybe 30 times more effective at trapping heat, even if it doesn't last as long in the atmosphere. It's a big deal for warming.
So the cow is just the container, you could say. The real workhorses are microbes inside, specifically a group called methanogens.
Exactly. And methanogens belong to this fascinating domain of life called archaea.
Ah, archaea. Distinct from bacteria, distinct from eukaryotes like us?
Right. People often call them chimeric. They look structurally a lot like bacteria, typical prokaryotic shapes, rods, spheres. But when you peek inside at their molecular workings, especially how they handle their DNA and make proteins, they look surprisingly, well, eukaryotic,
Right.
like us.
That mix is really what gets me. So our mission today for you listening is to unpack that. What makes archaea tick? What are these unique adaptations, molecularly, structurally, that let them survive where almost nothing else can? And how are they really different from bacteria?
Okay, so let's start with where they live. We used to think of them only in extreme places, boiling hot springs, super salty lakes, really acidic environments.
The extremophiles.
Exactly. And while many are extremophiles like those methanogens or salt-loving halophiles or things like sulfobis that love heat and acid, we now know they're actually everywhere.
Yeah.
Just the ones we can easily grow and study often are those extreme types.
And when we look at them under a microscope, they're shapes. You mentioned rods and cocci, like bacteria, but they get weirder, right?
Oh, absolutely. The diversity is incredible. You get branch filaments like in Thermoproteus tenax.
Yeah.
But my favorite example has to be Haloquadratum walsbyi.
Ah, yes, the famous postage-stamp archaeon.
That's the one. Just imagine a cell that's maybe 2 to 4 micrometers across. So tiny, but it's incredibly flat, only about 0.25 micrometers thick, like a little square sheet.
Okay, that's a wild shape.
Yeah.
Why be so flat? What's the advantage?
It's all about surface area versus volume. Being that flat gives it a huge surface area compared to its internal volume.
Right, more membrane exposed for the amount of stuff inside.
Precisely. So if you're living in a really salty place where nutrients might be hard to come by, maximizing that surface area helps you grab whatever you can find. It's a brilliant adaptation built right into its form.
So you've got these super flat ones maximizing their surface area, but then the reading also mentioned giant archaea, forming long filaments.
Yeah, things like Candidatus giganthama carucarens. These can form filaments up to 30 millimeters long that's visible to the naked eye, often covered in bacteria, suggesting maybe some kind of symbiosis.
Wow! From postage stamps to these massive microbial structures, the range is huge.
It is. And underlying all that diversity, the key to their survival, especially in tough conditions, really comes down to their cell envelope, the membrane and the wall.
Okay, this is where archaea really carve out their own niche. Starting with the plasma membrane lipids, they're fundamentally different from ours or bacteria's.
Two main things. First, the fatty acid chains aren't straight, they're branched. They're built from 5 carbon units called isoprene.
Okay, branched chains. And the second thing?
The second is how those chains are attached to the glycerol backbone. In bacteria and eukaryotes, it's an ester linkage. Think of it like, well, it's relatively easy to break, chemically speaking. Archaea use ether linkages. These are much, much tougher, more resistant to chemical attack, more resistant to heat. It's like molecular superglue compared to regular tape.
That makes perfect sense for extremophiles. If you're getting boiled or pickled, you need bonds that hold together.
Absolutely. And there's even a subtle difference in the glycerol molecule itself, the stereoisomer they use. It all adds up to a fundamentally different, more robust membrane foundation.
And this chemistry allows for different membrane structures too, right? Not just the standard bilayer.
Exactly. So some archaea have lipids called glycerol dieters. These have C20-20 carbon chains, and they assemble into a typical lipid bilayer, 2 layers thick, just like in bacteria.
Okay, standard bilayer. But there's another option.
Yes. The glycerol tetraethers. These are amazing molecules. They essentially link 2 glycerol backbones together with really long 40 carbon chains spanning the entire membrane width.
Whoa! So instead of 2 separate layers, it's one single continuous molecule running all the way through.
Precisely. It forms a lipid monolayer. Imagine one solid rigid sheet instead of 2 flexible ones.
That sounds incredibly stable.
Yeah.
especially against heat.
It is, and that's what you see. In extreme thermophiles, things like sulfolobus living above 85 degrees Celsius, their membranes are almost entirely made of these tetrathermal monolayers. It provides the rigidity they need to not just fall apart at those temperatures.
And that stability probably helps those methanogens in the cow's gut, too, dealing with heat and acidity.
Definitely. Now, even with these tough membranes, they still need to get nutrients across them,
Right. How do they eat?
mostly through active transport, similar to bacteria, using energy to pump nutrients in, often against a concentration gradient, because they might be living in nutrient-poor places. Primary active transport, secondary transport, sometimes even group translocation. Pretty standard toolkit there.
Okay, so unique membrane chemistry, standard transport mechanisms. What about the cell wall outside the membrane?
Right, the cell wall. Here's the absolute defining feature. Archaea do not have peptidoglycan. None.
That's the stuff that makes bacterial cell walls rigid, the target for antibiotics like penicillin.
Exactly. So Archaea lacks it completely. This means, by the way, that doing a Gram stain on an archaeon, well, it might turn purple or pink, but that result doesn't tell you much about its actual wall structure like it does for bacteria.
So it's not a reliable test for them. What do they have instead?
The most common thing is an S layer, basically a precisely arranged layer of protein or glycoprotein, like microscopic chainmail, right outside the plasma membrane.
And sometimes that's it, just the S layer.
Often, yes, it can be the only wall component. But others have different strategies. Some methanogens, for instance, have something called pseudomurein.
Pseudomurein sounds like fake peptidoglycan.
Kind of. It looks structurally similar, forms a similar mesh, but the chemical building blocks are different. It uses different sugars like N-acetyltyrosine acid instead of N-acetylmuramic acid, and different amino acids, L-isomers instead of D-isomers, in the crosslinks.
And those small chemical changes matter.
Hugely. They make pseudomurein resistant to things that break down peptidoglycan, like the enzyme lysozyme or penicillin. It's a clever molecular defense.
Built-in resistance. What else is out there?
You see other variations. Some have thick polysaccharide layers, like methanosarcina with its methanochondroitin. Some, like methanospirillum, have a protein sheath holding cells together. And then you have archaea with no rigid wall at all.
Like thermoplasma.
Exactly. Thermoplasma and ferroplasma, they just have their tough plasma membrane and maybe a slime layer, a glycocalyx. They manage without a rigid wall.
And then there's that really weird one, Ignococcus hospitalis. You mentioned it earlier. It has two membranes.
It does. It's got its plasma membrane, but then outside that there's a large space, an inner membrane compartment, and then a second outer membrane, very much like a gram-negative bacterium.
With pores in the outer membrane even.
Yes, protein pores that function similarly to bacterial porins. It's a structural setup that really makes you wonder about evolutionary history. Is it shared ancestry? Or did they independently arrive at the same solution, convergent evolution?
Fascinating. Okay, let's move inside the cell now, the cytoplasm.
Right. Inside, it looks broadly similar to bacteria. You've got ribosomes, DNA packed in a nucleoid region, various storage granules or inclusions.
Do they have a cytoskeleton?
They do. And interestingly, they have proteins that are related to bacterial cytoskeletal proteins like FTSE, which is related to our tubulin, and MiraB, related to our actin. But they also have unique archaeal components like crenactin and sadidzi proteins. So again, bit of a mix.
But the ribosomes, you said earlier, they really highlight that chimeric nature.
Absolutely. Size-wise, they're the same as bacterial ribosome 70S, made of a 50S and a 30S subunit. That's prokaryotic.
Okay.
But the molecules inside those ribosomes tell a different story. The ribosomal RNA, the 16S, 23S, and 5S RNA, while similar in size to bacteria's, have distinct nucleotide sequences. That sequence difference is actually one of the key reasons we classify archaea as a separate domain.
And there's more.
Yes. Some archaea also have an additional small RNA, the 5.8S RNA. You don't find that in bacteria, but you do find it in eukaryotes.
Ah, another eukaryotic link. What about the proteins in the ribosome?
More proteins than in bacteria.
Huh.
Archaea have around 68 ribosomal proteins. Bacteria have closer to 55. And here's the really crucial point for understanding their relationships and why many antibiotics don't work on them.
Okay.
Every single ribosomal protein that is shared between archaea and bacteria is also found in eukaryotes. There are no ribosomal proteins unique to just archaea and bacteria, but there are proteins shared between archaea and eukaryotes that bacteria don't have.
So the ribosome structure, protein-wise, leans more towards the eukaryotic side, even though the overall size is bacterial.
Exactly. That's why antibiotics designed to target bacterial ribosomes usually leave archaeoribosomes untouched. They're just different enough at the molecular level.
Okay, that's a key difference. Now, what about the DNA itself, the nucleoid?
Like bacteria, it's typically a single, circular, double-stranded DNA chromosome. But there's an interesting pattern with chromosome copies.
How?
Many Archaeota, one of the major archaeophylas, are polyploid. They keep multiple copies of their chromosome in the cell. Archaeota, the other major phylum, tend to be monoploid, just one copy.
Any idea why the difference? Polyploidy. Maybe that helps ensure each daughter cell gets a full chromosome, especially under stress.
That's a leading hypothesis, yeah.
Yeah.
But the most striking eukaryotic similarity in the nucleoid is the presence of histones in many archaea, especially Archaeota.
Histones, the proteins we use to wrap up our DNA into nucleosomes to compact it.
The very same, or at least very similar proteins, they form structures that look like eukaryotic nucleosomes, helping organize and compact the archaeal chromosome.
And why would they need that? Especially thermophiles.
Well, think about it. If you're living near boiling point, your DNA is constantly at risk of denaturing, basically melting apart. Wrapping it tightly around histone proteins is thought to provide significant thermal stability. It physically protects the DNA.
Structure enabling function again.
Yeah.
Amazing. Okay, last major area. Structures on the outside for moving around or sticking to things.
Right. Archaea definitely have external structures. They have peci, similar to bacterial type 5e pi used for attachment, maybe DNA exchange.
Like that UV-inducible PLS.
Exactly, the ups pilus and sulfolobus. When the cell gets zapped with UV light, which damages DNA, it makes these pili. The pili help cells clump together, aggregate, and it seems they then facilitate DNA transfer between cells, likely as a way to share genetic information for repair.
Clever survival mechanism. And they have even stranger attachment structures too, right?
They really do. Things like cannulae hollow tubes found on Pyrodictium species that actually connect the cytoplasm of adjacent cells, forming this extensive network.
A network of connected cells.
And then there are the hami. These are found on some archaea that live in biofilms. They look like tiny grappling hooks sticking out from the cell surface.
Grappling hooks? Seriously?
Seriously, they have a barb at the end, perfect for latching onto surfaces or maybe other cells within that biofilm community, clearly built for attachment.
Incredible structures.
Yeah.
What about getting around flagella?
They have flagella, but we call them archaea because while they function for swimming, they are fundamentally different from bacterial flagella.
How so?
Several ways. Archaea are thinner, only 10-14 nanometers thick compared to 1822 for bacteria. The filament itself isn't a hollow tube like in bacteria, and it's often made of multiple different types of protein subunits called archaeolins.
Okay, thinner, solid, multiple proteins. How are they built?
This is really interesting. They're assembled more like type 5e pili. The archaeolin proteins are added at the base of the growing filament, pushing it outwards. Bacterial flagella grow by adding subunits at the tip.
Completely different assembly. And what about the motor? What powers the rotation? Still proton motive force.
No, that's perhaps the biggest difference. Bacterial flagella spin using energy from protons flowing across the membrane, the proton motive of force. Archaea rotate using energy directly from ATP hydrolysis.
They burn ATP to spin, a
Wow.
totally different energy source.
Totally different engine. And the movement is simpler too. They rotate one way to go forward, reverse rotation to go backward. No complex run and tumble like bacteria.
And this ATP-powered motor, is it fast?
Incredibly fast. Some archaea, like Methanococcus villosus, are among the fastest swimmers known in the microbial world. They can clock speeds up to 500 body lengths per second.
500 body lengths a second. Why be so fast?
Think about where some of them live, like those deep-sea hydrothermal vents. You've got superheated water blasting out next to freezing cold ocean water. There's a very narrow zone with the just right temperature.
Ah, so they need speed to quickly navigate those steep temperature gradients to find their preferred spot.
Exactly. They use that speed to explore, and then once they find a good niche, those archaea can also function in attachment, helping them stick there. They also show taxis moving toward attractants or away from repellents like chemotaxis or even phototaxis in some halophiles, using signaling systems that are related to bacterial ones, despite the motor differences.
Okay, so wrapping this all up for you, the listener, what's the big picture on archaea structure?
It's a story of unique adaptation and really molecular blending. The absolute key takeaways are those unique membrane lipids, branched isoprene chains, stable ether linkages, allowing for those super rigid model layers in extremophiles.
Right, and the cell walls, remember, no peptidoglycan. Instead, you see diversity, S layers, pseudomurein, other polysaccharides, or sometimes no wall at all.
And then internally, that chimeric nature shines through. ribosomes with bacterial size but eukaryotic protein features and rRNA details, a nucleoid with bacterial-like circular DNA but often compacted using eukaryotic-like histones, especially in thermophiles.
Plus those unique external structures like hami and cannulae and the ATP-powered archaella, it's a whole different toolkit.
It really is. They've mixed and matched molecular strategies, leveraging stability of the tetraethers, the pseudomurein, and unique mechanics like the archaella to conquer environments where little else can survive. Understanding this structure-function focus is crucial to grasping the sheer diversity of life.
So, a final thought to leave you with. Given that archaea use things like histones and have ribosomal features that seem to bridge the gap towards eukaryotes, what else might we discover about them that could completely shake up our view of early life and maybe even that mysterious last universal common ancestor, LUCA? Something to think about.